Thank you for giving me the opportunity to talk. It's great to be here. So I'm going to give you an idea of some of the different work we've done on binding free energies, kind of a couple of things that are mostly done, some of which started at my postdoc and then have carried on as I started at UNO, and then a quick snippet, hopefully at the end, of what we're doing now. A big project we've got underway. And if I'm a little bit scattered, it's just because we just had some twins a couple weeks ago. And they just actually got home last Monday. So they're a little early. So great, yeah, twin boys is what we have too. So so that makes 4 kids now. Let me start off by saying. Talking a little bit about why we're so interested in predicting binding free energies. And so, what I'm going to be talking about here mostly is binding of a small molecule ligand to a protein, like we see here in this figure. We're focusing mostly on small molecules rather than protein-protein binding, for example. What could we do if we could predict these well? Among the things we could do are designing molecules to manipulate protein function in desired ways. We could do things like recognizing potential toxins by looking to see what proteins in the body a new molecule we're interested in might interact with. Right now I'm just imagining we could do these reliably and accurately as fast as we wanted. So some of these things might be a long ways down the road, but this would be what we'd be interested in. We could use these kinds of tools to help identify functions of enzymes. An enzyme has to be able to bind its substrate, so it could give us an idea of what an enzyme of unknown function could interact with. It could help us do things like protein design. Again, a protein has to be able to bind its target molecule. And here I'm mostly going to be focusing on aiding medicinal chemistry, that is, aiding the drug discovery process. There's a whole lot of things that we could do, maybe finally enable cold fusion, perhaps not, but the point is there's a lot of things that accurate binding predictions could help us do. Now, the challenging thing about binding free energies is that they involve a ratio of partition functions. Binding free energy is this equation. These Q's are partition functions, and each of them is for a different system. QPL is for the protein ligand system in whatever solvent it's in, QP for the protein alone, and QL for the ligand alone. Each of these is solvated, of course. And these things aren't just from a single snapshot of the system. Really, each of them comes from a weighted integral over all of the relevant configurations of the system, and there may be multiple relevant configurations of the system, and those configurations can be different for the ligand in the complex, for the ligand alone, for the protein alone. So that sort of presents a challenge. You don't get a binding free energy just by looking at a PDB structure of a ligand and a protein. It involves a whole bunch of configurations potentially, and it's a weighted integral. Yeah, the question is, do you have to put a limit on the number of configurations you use? Practically, yes, because you can't count everything, but that's, a question is, well, how many do you need to use? And so that's not really a resolved question. It's just important to remember that it isn't something that comes from only one. It comes from potentially a semi-infinite number of them. And that's part of the challenge, why binding prediction is so challenging. One of the main methods that's used in pharma for trying to predict binding to some extent is docking methods. Docking methods basically are a shape-based way of trying to predict which molecules will bind well. And these are just some pictures to show that docking, while it works well for some things, doesn't work well at predicting binding strength. On the vertical axis you have docking score, which is something that's supposed to correlate to some extent with how well molecules bind. And on the horizontal axis, you have binding free energy. And you'd like, a nice linear correlation between these two that would allow you to use your docking score to predict how well something binds. And basically, these are just some representative plots from an extensive study, and they kind of all look just like scatter plots. There's not really any correlation. And this is showing you that docking doesn't work reliably to calculate binding strengths. It does work for some other things, but it doesn't work as a measure of predicting binding strength. But this is one of the most commonly used methods for other reasons I could talk about later if anybody's interested. In fact, here if you look at the field of binding prediction, docking is basically most of it, partly because it's simple, it's fast, and there's very few good alternatives. There's another piece of this pie, which is what I call approximate physics-based methods. They still don't evaluate binding free energies, that is, that ratio of partition functions I talked about. Docking doesn't do that either. But they add a bit more physics in. This orange piece of the pie here is rigorous physics-based methods that do evaluate binding free energies, but they require a structure of the ligand you're interested in the protein receptor, or at least a related ligand in the protein receptor. And so that's not necessarily always the case when you're trying to predict binding of a new molecule, perhaps in advance of making it, because you haven't made it yet and you don't know how it's going to bind. So where I've been focusing in is this tiny little piece here, which is relatively new, which is basically, let's try to predict binding predict really the binding free energy without knowing in advance how molecules bind. So we're doing the same kinds of things as in this pie, but we have this additional challenge that we don't know the binding mode in. We have to work harder to get that. So in my postdoc work, we started looking at this model binding site in T4 lysozyme. This is a really, really simple binding site. It's a design binding site. It's created by a point mutation, and it binds things like benzene and toluene. You could think of this as one of the simplest possible binding sites in some ways. And the hope was that we would just do a quick study in this binding site and plunge ahead to biologically interesting things. And that ended up not to be quite what happened. We're interested in this for a number of reasons I'll just go through really quickly. One of the most important ones is it's well characterized experimentally. It's easy to get new data. And that gave us the opportunity to make blind predictions, which is a great test and challenge. It's always harder to get the right answer when you don't know it in advance. Or you can avoid fooling yourself that way. And hopefully anything we would learn in this binding site would carry over to more biologically relevant binding sites. So we hoped it was a good, simple starting place. The way we approach all of what I'm going to be talking about basically is using a thermodynamic cycle to compute binding free energies. And this is based on molecular simulations, particularly molecular dynamics simulations, so it involves a lot of simulation. We're interested in the free energy of taking the protein plus the ligand and combining them to the complex. And simulating those association and dissociation events is sort of challenging. So we instead take a cycle where our particular approach involves restraining the ligand first to a reference orientation in the binding site, for technical reasons I could talk about later. And we compute the free energy of doing that. Now we turn off the interactions between our ligand and the protein. You can think of it as making the ligand into a stealth ligand. It doesn't interact with the protein anymore, and the protein doesn't interact with it. So now if the protein had changed shape to accommodate the ligand, it should be able to change back. We compute the free energy of doing that. And this is something we can only do in the computer, of course, because you can't make a stealth ligand in reality. That's now equivalent to having our protein with no ligand there, and our restrained ligand basically by itself. We then remove the restraints, and that's equivalent to having our restrained ligand in water. And then we compute the free energy of turning back on interactions in water. And if we've done these right, if we've invested enough simulation, we've computed these free energies properly, the free energies around here, around the bottom of this cycle, will equal, the sum of those will equal the negative of the binding free energy. So that's the idea. That's not my thermodynamic cycle, but everything that I'm going to be talking about from now on is stuff that I've done. All right, so the big challenge here, though, is you may have noticed that I was sort of starting with something like the bound structure, and I've already told you that I don't want to have to know the bound structure of the ligand in the protein. So we need to predict that somehow, and our starting point for this was basically to take a bunch of potential orientations of the ligand and just stick them into the unbound structure using docking, which is this method I discussed at the beginning. It's good at generating things that are reasonable. And then since we're going to be doing simulations, hopefully our simulations will find the right binding modes. And that was our initial hope, but it doesn't always work that way. And my movies are broken because of transferring this between laptops. What you should see here that you don't is that on one side you've got a ligand wiggling around in one orientation in the binding site, and on the other side you've got the ligand wiggling around in another orientation in the binding site. And those the ligand never flips between those two different orientations in our simulations. And that tells us basically we have sort of a problem when we're trying to predict which binding mode the ligand has. when we stick it into the binding site, it isn't just going to find the right binding mode. Often, in fact, it's just going to stay stuck where it started. And how then do we know the right binding mode? Well, the way I came up with to deal with that was to, instead of just trying to find the right binding mode, what we can do is compute an effective binding free energy for many different orientations of the ligand, like delta G1, delta G2, and there's a rigorous way to combine those. So we don't have to know the binding mode to begin with, we just have to find every potentially relevant binding mode and cover those. Another problem we ran into as we worked on this was that ideally our simulations cover all of the relevant motions of the protein. Potentially as the ligand binds, the protein could change its shape or conformation somewhat. And if our simulations cover those motions, we should get results, binding free energies, that are independent of what structure the protein started in. Here we've done, this is p-xylene bound in the binding site. Here we've done the binding free energy calculation from two different structures of the protein. Apo is unbound and holo is bound. And what I just said is that these two numbers, the binding free energies we compute, should agree if our simulations are long enough. And they don't agree, obviously, and the experimental value is in between. And if you look at this figure, this is using some of the structural data that's available. Green is the unbound structure of the protein, without this pink molecule here, and pink is the bound structure of the protein. So the only significant change in the protein that's happening on ligand binding is this single side chain, this valine side chain, reorients on binding of p-xylene. And if we look at what happens in the simulations, we never see this side chain reorient during our simulations. Basically, it's too slow for our simulations to sample this motion, and so our results depend on where we started. So once we know that's the problem, we sort of have a workaround, which is to compute the free energy of reorienting this side chain and include that free energy in the binding calculations. Then we can get consistent results regardless of what structure we started from. And that agrees better with experiment than before. The key here is that we got consistency. If we're doing everything right, we should be able to get consistent results regardless of what structure we started from. Okay, after addressing these challenges, we ended up at a point where we could get calculated versus experimental binding free energies for this binding site that worked pretty well. The vertical axis is calculated, horizontal axis is experiment, and you want to compare this with those plots of the docking results for a bunch of binding sites that I showed earlier that look just like scatter plots. This isn't great, but if the results were exactly right, they'd lie along this line. So there's some kind of a correlation and clustering around this line here, and our RMS error is about 1.8 kcals per mole, which is not necessarily as good as we'd like, but it's a lot better than it could be. So this work was where we already knew the answers. And so the real test is whether we can make predictions. So we took some small molecules that our collaborators and docking methods thought should bind, and we predicted their binding free energies and then had them tested experimentally. And so the calculations were done without knowing the experimental results and vice versa. And so here we predicted negative 5.6, we got negative 6.4, so off by 0.8, 0.7, 0.1. These last two cases, they just put bounds on the binding free energy. They weren't actually able to measure it with calorimetry, but we were consistent with their bounds. We also did some binding mode prediction stuff on these molecules, and that worked out quite well, but I won't talk about it because I want to move on to more recent work. So this actually worked out better. The predictions worked out better than we even hoped. This was quite good. But this is a really simple binding site. So more recently, we moved on to a polar version of this binding site. It still binds some of the same molecules. It still binds toluene. But now we've introduced an additional point mutation, which adds the possibility of hydrogen bonding. So the original binding site doesn't bind phenol, and this one does. So it's another step toward the biological binding site. And we did much less retrospective work here because there was a lot less experimental data that was already available. So I'm going to tell you more about what we did when we did predictions. And I'll throw a whole bunch of data up there, kind of sorted by category Here's again the binding site. There were a number of cases where predictions worked out really well. Here's what some of those molecules look like. This category is basically stuff where we're off by less than 1.5 kcals per mole. So, and in some cases the experiment didn't get a number, they just got a rough idea. But those looked pretty good. There were some cases where we correctly recognized that they don't bind or bind very weakly, like these. What's more interesting, I think, is when we don't do well. Here's a couple where we just got pretty close. The error was a bit larger than we would have liked. Here are some where we know, I think, what went wrong. Here, this last column again is the error in kcals per mole. In this first one, there was a water that wandered into the binding site that shouldn't really have been there. But because it was sort of as a fluke got stuck in the binding site, it was trying to occupy the same space as the ligand, and it made it look like the ligand didn't bind well, when in fact if the water wasn't there it would bind better. So sort of a statistical fluke. Occasionally if you're doing a simulation something can happen that shouldn't really happen. If you were able to run your simulation long enough, but because you didn't run it long enough, your results end up bad. And you don't know that in advance. This next case, we just didn't find the right binding mode of the ligand. So if you don't find the right binding mode of the ligand, you can't get the right binding free energy. These last two cases, there was a slow motion of the protein that we had never seen before that suddenly happened. There's a partial unfolding of this helix, helix F, going from the green to the light blue here. And that creates more space for the ligands. So if you don't have that motion, your binding free energy estimates end up too positive. It looks like the ligand doesn't fit. If you do have the motion, it would end up more negative. It's a slow motion, so it's been really difficult for us to sample that in our simulations. There's some cases also where we just don't know what went wrong, and we're following up on that. I want to take you through one little success tidbit from this that I thought was particularly interesting. This molecule, thiophene 2-carboxyl dioxide, the calculations initially said it should bind well, and the experiment said it would be a non-binder, or it is a non-binder or weak binder, which was perplexing. We couldn't find anything obviously wrong. Then when we looked more closely, it turns out that there's two isomers of this molecule, and the calculations had sort of included both because we weren't really paying attention, and we just plunged ahead without noticing we had two of them. And the experiment was done on 98% pure 1. So we're comparing apples with oranges a little bit. Experiment is saying 1 doesn't bind, and calculations are saying that 1 or 2 binds. So while we were figuring that out, the experimentalists actually followed up and tried to do crystallography to confirm that 1 really didn't bind. And so they took 1 and they soaked it into the crystal, and lo and behold, they found something binding, but it was 2. And so how did that happen when they were doing experiment on number 1? Well, apparently isomer 2 is present as a 2% impurity in the solution of isomer 1, and isomer 2 binds well enough that it pops into the crystal structure when you do that. So, and it turns out, looking back at the calculations, that we were saying isomer 2 binds. So, in fact, experiment and calculations agree here, when it looked like initially that it didn't. We also did a number of binding mode predictions here. I'm showing you just a couple of them. On the left is the electron density with the modeled structure, and on the right is the electron density with the deposited structure. And on the right is that structure with the green being our prediction. In this case, we did quite well. In this case, we've got the ring, in this bottom case, we've got the ring position right, but the intermediate position of this tail is not correct, which is kind of interesting. We're doing a lot more work to follow up on this still because we think we did have some sampling problems here that we have some better algorithms to fix now. One of those challenges is this, that when you introduce hydrogen bonding in this binding site, it creates more potential ligand binding orientations, more metastable orientations. Here are some of them for one of these molecules. Now we've got about 6 or 7 of them. And this picture was going to recapitulate my movie that I wasn't able to show you, but basically here are 2 different orientations we were considering. We already know in principle how to consider multiple potential orientations, but when you start up ending up with this many, 6 or 8 or 10 or 12, you just need better ways to separate them and account for them. And so we've had to develop a kinetics-based way to separate orientations and combine them. Let me just skip over that and tell you a little bit about what we've learned here. I can come back if there's time. So one of the things we've learned is that there can be big kinetic barriers between orientations of even pretty small ligands like these. These are probably like fragments of biological ligands, meaning that it takes a long time to get from this orientation to that orientation. And so what we end up needing to do is consider multiple candidate orientations, because the simulations aren't going to find the binding mode for us. We also found that we need to use kinetics to separate potential ligand orientations. We found that even small conformational changes in the protein, such as a side chain reorientation, can present kinetic barriers, but those can be worth a couple or a few kcals per mole in the binding free energy. That I didn't talk about. We still need to do a better job of sampling things, especially side chain motions. And yet already we can make non-trivial predictions like what isomer this molecule binds. And hopefully I've convinced you that we can systematically track down some problems and make improvements. Let me wrap up by telling you really quickly about something we're working on now, which is trypsin, which binds, it's a serine protease, so it's important and well studied because it's a member of this serine protease family, of which there are many targets which are important for drug discovery. It's also easy to get it and easy to do structural studies on it. There's currently a blind challenge going on to predict binding of a whole bunch of potential new trypsin inhibitors. Somebody's experimentally screened the Maybridge fragment library, which is about 500 compounds, for binding to trypsin. They have affinities for the compounds that bind and crystal structures, and these are not being released until after predictions are submitted. So we're involved in that. And this is benzamidine bound in trypsin, which is one of the known inhibitors. This is a big challenge, and so I said that. We have to predict this by June. And there's really 2 stages to this that we're trying to do. One of them is called virtual screening, which for those who don't know the lingo just means let's take all of those compounds and predict all 500 and predict which ones bind. Then the second stage is of the ones that actually bind, predict how well they bind. And we're trying to just do the whole thing, meaning let's actually just predict for all 500 molecules which ones bind and how well they bind. And this is kind of a crazy map that's hard to see, but basically what we're doing is clustering all the molecules structurally, which are these little dots up here. Each dot is a molecule. And then we're doing a giant map of calculations comparing the different molecules. That will allow us to get the relative binding affinities of all of the molecules. So there's about 500 molecules. We're planning about 1,100 calculations between molecules. We're going to do each of those 10 to 20 times. So it's 20-some thousand calculations, and we're planning to do that unfolding at home. So that's getting underway right now. And obviously we have a lot of challenges dealing with the massive amount of data we're going to be generating there. Anyway, so let me put up my acknowledgments, and thank you for your time. Fareed. I didn't talk about charged molecules. We have, well, the trypsin ligands actually are charged, typically. There's some probably that aren't, but benzamidine is charged. There's some unique challenges in dealing with binding free energies of charged molecules that actually mean that for now we only do relative binding free energies of molecules within the same charge. We've worked a lot on trying to compute absolute binding free energies of charged molecules, but we don't fully understand all the issues there yet. Anybody else?
I'll just throw in a quick question. So I use using folding at home. What's the individual size of these simulations?
They're not that big. I think it's about a 35,000 atom system.
Can you do a back of the envelope calculation of how soon your end-to-end solution would go through versus using folding at home versus TerraGrid with a dedicated application?
No, partly it's just a allocation. Well, for one thing, there's a data management issue that's easier to deal with if we're on computers that we fully control, it seemed like to us. And then the other is that there's the writing the allocation, submit the allocation, wait for the time to become available kind of issue. And we can just start these running as soon as we're ready. So not really. Yeah, that's the answer. Not really.
It's very interesting. This last answer about the barrier to get time on partition. That's a very well-known problem. I'm glad to hear that. That's useful feedback.
The other big problem we often have is these are individual simulations which we need to run for a relatively long time. And because we have so many of them, it's silly to parallelize them because we have 50,000 simulations to run. So we run into wall clock time limits. Like we can send these off unfolding at home and let them run for 2 weeks, an individual simulation run for 2 weeks. And we can't do that on more typical shared clusters. Run into 48 hours or 72-hour time limits.
There are techniques to self-spawn when it comes to an end with the wall clock time. We can talk about that. So exactly, that is the problem. Individual simulations are 1, 2 cores A, B. They need to run for way longer than the typical queuing time assigned. So cluster them, and then you have them self-checkpointing and self-migrating as and when we saw.
Yeah. Yeah, so I knew that there was a way to do it with those tools, but given that we've got from February to June, it was simpler to basically proceed.
I'm asking you because you're up against a challenge, so trying to get this done in a finite amount of time is an important consideration. And I can understand why folding at home in general would be easier than doing it in TerraGrid for this class of problems. But you've added this additional constraint that you're in a challenge and it's like the CASP. You've got to get it done by a certain amount of time.
Right.
And then to hear that you did not go for the dedicated cycles versus try the folding at home approach is very interesting.
Okay, thanks.
Okay. Well, thank